Want to live forever? Many people have tried. Only their images remain. Evidence that for thousands of years people have wanted to find the key to life. They sought it in monuments. They sought it in faith. They even tried magic potions. In 1839, a Dutch chemist, Gerardus Mulder, discovered a compound that he believed to be the key to life. To reflect its great importance, he named it after the Greek word proteus, meaning first or primary. He called it protein.
Mulder was investigating animal products like albumin from eggs, gelatin from skin and bones, and fibrin and globulin from blood. He got the idea that one basic formula was common to them all. He thought protein, as he called it, was a particular big organic molecule. He was right that protein is important, protein's in every living thing, but he was wrong about the molecule. He'd actually found a polymer, which is a molecule made up of a number of smaller units.
In fact, the small units in one protein, gelatin, had already been found by Henri Brassenot. Brassenot, remember, was the man who boiled up sawdust, which is mainly cellulose, and discovered that it was composed of glucose units. He then tried the same trick with gelatin, and likewise got a deposit of a sweet crystalline substance. This would later be called glycine, from the Greek for sweet. But he knew it wasn't glucose because he could get ammonia from it.
Now, ammonia is a compound of nitrogen, and only a few of the other substances in the body have nitrogen in them. When proteins break down, they generate ammonia. So all living things have to find ways of getting rid of ammonia, which is poisonous. Humans get rid of it as urea, hence the ammonia smell of urine when the urea in it breaks down. And some of it may come from glycine.
Here is glycine, and if I put some ordinary table sugar, that's sucrose, next to it, it looks just the same, and it tastes sweet too. But unlike sucrose, glycine contains the nitrogen functional group NH2. Compounds with NH2 are called amines, and they are bases. But glycine also contains the COOH functional group, which also makes it a carboxylic acid. So this stuff spans two families. It's partly acidic and it's partly basic. It's in a group of its own. It's an amino acid. I know it doesn't look like much, but amino acids are the key to life.
Glycine is the simplest amino acid. Its molecule is simple. On one side is the amine group with its atom of nitrogen. On the other is the carboxylic acid group with its double bonded oxygen and its OH tail. But from this rather plain looking combination, nature expands into a dazzling array. First, it creates about 27 variations on the glycine molecule. As we've seen before, it's common to substitute other atoms for a hydrogen on a carbon chain. It may be as simple as a methyl group which changes glycine into alanine, or as complex as the double ring tail of tryptophan. In humans, the variations are less, amounting to a total of only 20 different kinds of amino acids. Nature, it seems, can do all it needs protein-wise with these 20 molecular building blocks. It doesn't vary the glycine base, and it even chooses to join on all the variations to the same place. Thus, if all the variations are indicated by the letter R, we can see the straightforward family formula of general amino acid structure. But there, the conservatism ends. Amino acids can be induced to do a kind of acid-base self-neutralising trick. As their molecules come together, a molecule of water drops off and they bond. And bond, and bond, sometimes into chains thousands of units long. life spins fantastic polymers of protein. This is stuff we all know well. It's muscle. It is skin and scales, tendons, cartilage, blood and vital organs. It's us.
There's a huge difference between chemicals like these and a living thing. But it turns out the difference is not one of kind, but of complexity. Here's a 3D model of glycine. Here's the carboxylic acid group, and here's the blue nitrogen with its two hydrogens, the amino group. But it's here, in the place of one of these central hydrogens, that all the other side groups attach. The 19 other variations go on here. But that's simple compared to when you start linking them up. I've just made up a basic model of a chain. Here's the spine and these bonds where the 20 variations go. For instance, you could have a glycine here, an alanine here, a cysteine here, a leucine here, and so on. Now this is only 7 units long, so it doesn't qualify as a protein. Proteins are much larger. This is referred to as a peptide, but you're starting to get the picture.
The number of possible proteins is literally endless. Take a tiny little peptide, let's say 4 amino acids in a chain. With 20 varieties to choose from, that's 20 raised to the power of 4 ways to combine them. 160,000 ways to assemble a 4-unit peptide. When you get to a largish protein, say 500 amino acids in a chain, the number has 600 zeros in it, and every one is a possible protein. Of course, we know that they can't all exist, because that number is larger than the number of atoms in the universe.
As if sequence isn't enough, there's also shape. Take silk, which is the protein fibroin. Silk is a straight chain, mostly made up of glycine, alanine, and so on. These long strands link up alongside one another to form a strong fibrous sheet. Compare this to hair, which is the protein keratin. Keratin, a completely different amino acid sequence, is coiled in a helix. This gives hair more stretch than silk. And these are just 2 shapes out of an amazing array that proteins can have.
Coils can coil around coils. Coils can coil over themselves. Chains may weave to and fro and cross-link to make sheets. Chains and coils may clump together in confusing tangles. Entirely different chains may associate in special interlinked relationships. In each case, they lock into position as a unique, stable chemical compound, sometimes with molecular weights in the millions.
Everything about a particular protein is important. For example, change its shape, even a little bit, and it simply won't behave the same. And some are quite fragile too. Take the albumin in egg white. Straight out of an egg, it's clear because it's in solution, but all you've got to do is whisk it or heat it, or put a drop of alcohol in it, and it comes right out of solution. This is an example of denaturing a protein. You've changed the shape of this amazing molecule, and now it behaves quite differently.
While proteins give life its structure, even bone has collagen and elastin threaded through it. For the most part, protein's role is much more complex. To maintain life, a vast array of chemical reactions must happen with great precision. The single biggest mystery is how even the simplest cell manages to perform all the hundreds of chemical reactions needed to stay alive at a high enough speed to be useful. Life is a chemical virtuoso, and the question is, how does it do it? The long-held suspicion was that the cell manufactured catalysts. A catalyst is something that can speed up a reaction without being used up itself. A biological example is making wine or beer with yeast. The yeast cells put out a substance that causes sugar to be converted into alcohol. The sugar is said to ferment.
When scientists first studied yeast, they used the term enzyme, which means in ferment. Then, when other biological catalysts were found, the term enzyme stayed on. For example, there's pepsin, which breaks down meat, lactase, which digests milk, invertase, which breaks table sugar into the simpler sugars the body can use, and the list goes on and on. And then people started to ask, what kind of chemicals are these enzymes, and how on earth do they control so many reactions in the body? It wasn't until 1930 that we got the first part of an answer to that question. What type of chemicals are these? Of course, you can guess what they found. Protein.
It took until the 1950s before a picture emerged of how proteins as enzymes worked in biochemistry. From the endless range of possibilities, nature has found some 10,000 proteins which fit their reacting substances like a lock fits a key. Attracted to sites on their appropriate enzyme, the substrates, as they're called, are brought close together. This drastically lowers the energy barrier to start them reacting, which they do almost instantly. The new substance formed pops clear of the protein, leaving the same site ready for more substrates to use it. Enzymes can speed up reactions 1,000,000 times or more. They are true catalysts because they are not used up in the reaction and very small amounts work quite well. Most are so specific for their substrates that they do not interfere with other reactions, even in the same solution. The result is a highly complex, coordinated chemical activity. Of course, while understanding this goes a long way to understanding the key to life, we must also ask, where are enzymes produced? How is it done? And how are they regulated to the right amounts? Where do we get our protein from anyway?
You have to have amino acids for proteins, and there are about 8 that we have to get from our diet. These are the so-called essential amino acids. The rest of the 20 we can synthesise from other nutrients. Meat and eggs, for example, are the most efficient sources of the essentials. But as we said before, proteins are a part of every living thing, so a selection of plant foods is fine also.
As for regulation of the enzymes, this is the job of hormones. If enzymes are the accelerator that makes life possible, then hormones are the foot on that accelerator that speeds life up or slows it down. As for where and how the enzymes and hormones are produced, the answer is yet more layers of chemistry. But ultimately it can be asked, what in the very first instance sets all this molecular machinery in motion? For some time, scientists suspected that the ringmaster would be another kind of protein. But they were wrong. The control agent sitting behind it all, finally discovered in the cell nucleus, was not a protein. Proteins are the key to life, but they are not the locksmith who designs it. That's another story.